be from any source, especially in river systems.
Any other questions? Okay, then we're done. Thank you so much, Dr. Botha. Okay, then up next we have Dr. Joe Leroy again. He has already been introduced by Scabot, so we just welcome him back onto the stage. Thank you.
Thank you very much. It's always a bit difficult to speak after lunch because everybody is having a postprandial dip, as we call it, myself included. So please don't bother. Use your time to relax a little bit. Don't snore. That's not so social. I'd like to take the opportunity to thank VetLink. They are great in organizing conferences. That's what I heard. I see it here. But as a speaker, I truly feel well taken care of. It's incredible. It's incredible. I travel a lot for lectures and meetings, etc. And honestly, there's nothing that can reach that level of VetLink. So to Madeleine and Kaylee, bye donkey. I can give my talk in Dutch if you prefer. Probably most of you will understand. Dus vandaag gaan we praten over fertility in dairy cows. They do deserve a long-term planning. And I will use the oocyte as our red thread in that story. I have to admit that dairy cow settings, the dairy cow farming industry in South Africa is different compared to European situation, and especially compared to the Dutch and the Belgian situation. I'm not from the Netherlands, never say that because you can make me angry there, but I'm a Belgian. I'm from the north part of Belgium, so the Flemish-speaking part. And there our herds are quite small, 100, 150 on average. So you understand that when it comes to fertility management, we are in a completely different setting compared to the South African setting. Nevertheless, I'm convinced of the fact that some of the concepts I will present today may be of help in your day-to-day herd health advice. So this is the most beautiful cell on earth. It's the oocyte. It's the biggest cell in a body, surrounded by cumulus cells. So you see these cells supporting the oocyte. It's fantastic. I could speak hours and days only about this fantastic juicy cell, but I'm not going to do that. I'll try to use the story of that cell to tell the story of fertility management in dairy cows. When are farmers starting to think about fertility management? That's at around the period of breeding, when we want to inseminate a cow. If you want to fall asleep right now, just wait for 10 seconds more. The only thing you need to understand or keep from this presentation is that if you only start thinking about breeding cows at the moment of breeding, you're far too late. So fertility thinking is thinking in advance, planning in advance, and taking care of the oocyte. So yes, if we want to have a pregnant cow after the voluntary waiting period, we have to have a conception. I hope you agree. We have to AI the cow some days before or some hours before. But before we AI the cows, at least in our conditions, we need an estrus. That's not the case here in most of the farms. We don't regularly off-sync cows. Important, if we go back in history and just be some earlier weeks after calving, we need a good ovarian, a good active ovary. That's very important. Without a nice resumption of ovarian activity, there will be no healthy oocyte at the moment of breeding. And to have an ovary that is nicely recovering, we have a recovered, or we need a recovered uterus as well. An inflamed uterus that is not well recovered after calving will send out signals in terms of inflammation, you remember from this morning, and will keep the cow infertile. No discussion about that. I will not cover uterus anymore in this talk. So in this talk, we will speak about the importance of the moment of breeding. What can we do with the oocyte at the moment of breeding? So I'm really thinking or speaking or focusing about the period around breeding, when we want to breed the cow. Second part of my talk will cover the balance between energy balance, negative energy balance, inflammation in relation to resumption of ovarian activity. And you still remember one hormone which was IGF-1, right? Oh, wow, it was really convincing. IGF-1. Before, and third in my talk, I will speak about the history of the oocyte. It takes about 3 to 4 months before an oocyte reaches the status of ovulation. So what happens with the oocyte before may have a carryover effect on the oocyte quality upon breeding. So that's what we're going to speak about. Okay, first, the moment of breeding. We have a juicy follicle here, ready to be ovulated, sprouting amounts of estrogen in the bloodstream, making the cow showing heat symptoms. That's the moment that we have to breed the cow, and then we have fertilization, which is taking place in the oviduct, by the way, and then at day 5, the embryo arrives in the uterus, and hopefully everything goes well, and we have pregnancy. Right. So there is one condition there. We need to have active ovaries, otherwise there will be no dominant follicle. And we need a cow in heat. However, don't forget, being an estrus is a cow physiology matter. Able to show estrus is a stable matter. If the floor is too slippery, cows are not going to show heat. And correct estrus detection is a man factor. So breeding cows on a heat is a joint venture of the cow, the management quality, the man, and the farm, the hardware. So don't forget it. Never blame the cow only. Inducing an ovulation, it's a cow matter. Timing and quality of AI is a man factor. And complete estrus recovery is also a cow factor. Uterus recovery is also a cow factor. So you see, it's really a joint venture. Never blame the cow only. Never blame the farm only. Just think about the combination. So what I can say, based on a lot of research we did in our laboratory, is that the oocyte and the follicular cells upon ovulation are very sensitive to their microenvironment. And I will give you some sneak peeks. So these are, I don't know if they are people, I used to be the president of the European Society of Embryo Technologies. I don't know whether they are ET people, embryo transfer people in the room, or ovum pickup people. No, this is an embryo, day 6. Early blastocyst. And I hope you see the difference. Left, pale and juicy, healthy embryo. Right, a dark embryo full of lipids. The dark embryo contains about 60% more triglycerides. It's just mayonnaise in that embryo. That embryo is flushed out of a dairy cow on a high-producing dietary regime, producing lots of milks. This embryo is also flushed from a dairy cow on the same farm, but that cow was just retired. She was happy on the pasture, absolutely doing nothing. So you see, just by looking at the embryo more in detail, that embryo tells me a lot about the mother that she or he is originating from. So that's super important. By the way, we see the same in human. We do similar work in human, exactly the same. If you want details, juicy details, I'm happy to share them, but after this talk. We do a lot of research in our laboratory. We have a farm or a stable next to the lab where we do ovum pickup. It seems that we only have female students, which is true. We do visualization of the follicles. We do follicle aspirations. These are not my fingers. I hope you agree. And then we do collect the follicular environment. We study what happens in the environment and relate it to the health of the cow or the diet of the cow, mineral composition, etc. So that's what we do. So for example, here, I hope you agree, 3 samples of 3 different cows, follicular fluid. 2 of them were given beta-carotene and 1 not. And you can just tell from the appearance of the follicular environment. So science can be very easy. It never has to be very complicated. So what else did we do in the laboratory? We wanted to understand what the impact of high NEFA concentrations were on oocyte quality. You still remember the NEFAs, the non-emulsified fatty acids? If you have a cow mobilizing a lot of fats, or having a lot of stress, or having a lot of pain, you have a lot of fats mobilized, and these fats ultimately arrive in the follicular environment. And we wanted to know what is the consequence for the oocyte. So what did we do? We went to the slaughterhouse. We collected immature cumulus oocyte complexes and we matured them in vitro for 24 hours. So we mimic the last 24 hours of the maturation process taking place in the follicle. And we added these fatty acids at relevant concentrations of cows being in pain and stress, whatever. So we call it the control group, those that matured in healthy conditions and those that matured in the lipotoxicity conditions. That's basically what it's called, the lipotoxicity conditions. Fertilizing and culture conditions in the laboratory were just standard. Same for all the groups. Just, again, only the maturation phase differed. Healthy environment versus stressed, lipotoxic, high NEFA environment. And then at day 7.5, we collected the embryos and we saw that in the lipotoxicity group, much lower amount of blastocysts were formed. So the oocyte suffers in terms of developmental capacity after fertilization, right? But there were healthy embryos formed. Some of them were healthy, and we decided to transfer the embryos in controlled recipients in a crossover design. And then we wanted to know if an embryo, after such a lipotoxic history, still survives until, say, day 7.5 and still looks acceptable, is her future also guaranteed, yes or no? So that's what we did. We transferred the embryos in groups of 8, and we looked a week later after flushing out these embryos. And normally after flushing, an embryo should look like this. Like this. It's like a spaghetti. We call it tubular embryos. They should grow aggressively like a tumor, basically, at 40 days of age. If they are retarded in growth, you have these more ovoid, like egg-type blastocysts or embryos. They are retarded in growth, and this one is really retarded in growth. So we flushed out the embryos, and we compared embryos originating from an oocyte that was matured in controlled environment versus those that were matured in lipotoxic environment. And so you see 2/4 of the embryos were tubular in the control group, only like 40% of the embryos that had an oocyte history of lipotoxicity, of stress, of high NEFA were able to develop through. We also did transcriptomic epigenetic analysis. I'm not going to bother you with that, taking the hour of the day, but it's incredible what happens in these embryos. Those that do survive until day 14, they look completely different. They behave completely different. Anyway, so we know from data in our laboratory and other speakers that all these factors, I only spoke here a little bit about NEFA, but there are many more. I hope you recognize LPS and TNF-alpha, adipokines, oxidative stress. They all have a drastic impact on that microenvironment of the oocyte. So around breeding, if the cow is lame or still suffering from I don't know what, the oocyte can tell, and you can expect reduced fertility rates. But there is more. We need, before we breed the cow, weeks before, we want that ovary resuming activity again. It's very nicely shown in research done in the late 80s and early 90s that the more ovarian cycles a cow can display before she reaches pregnancy, the higher the chance will be that she take in a calf successfully. So we call it the priming of the uterus. Uteri are primed by the precedent ovarian cycles, right? So that's important. So how can we make such an ovary active again? Well, here is the ovary. You all know this graph. I started this morning with that nice SIVA scheme, where you see how the brain communicates with the ovary and the uterus and the feedback signals, etc. It's basic physiology. So that's just here. The follicle grows, but look at this. I added adipose tissue. I added the liver. Look at this, IGF-1, growth hormones. There we are. I added pancreas because we need insulin. If we don't have insulin and IGF-1, the key to the fertility door will stay closed. Very easy. Every study confirms it. We need IGF-1, we need insulin to open the door to fertility, at least in the female part of the reproductive courses. So you recognize this part, right? I will go through it. For those who want to take a picture, you need to take the picture right now because now it's complete. I got the question. That's why I'm just showing it up again. There is a lot of interrelation, but what I wanted to show you is that if cows are in too negative stress and insulin is too low, you have massive mobilization, what I explained this morning. And again, we have a liver not able to do its primary task, support fertility and support milk production. So you get a high growth hormone cow and a low IGF-1 cow. What does high growth hormone? Boosting milk, boosting fat mobilization, but not helping IGF-1 because we have an uncoupled liver. The liver is not able to respond on the growth hormone signal from the brain. 2 reasons, I hope you still remember. The liver needs enough insulin to be able to put growth hormone into IGF-1. There was a second reason. If we have inflammation, IL-1, IL-6, too many necrosis factor alpha, also uncouples the growth hormone IGF-1 axis. Okay, so suffering cows are cows that are low in IGF-1 and low in insulin. So the fertility door stays closed. You can put loads of hormones. Don't do that. Give the money to me. It's much more well invested. Anyway, so a lot of indirect effects in that pathways, indirect affecting fertility. I'm not going too deeper there. We can discuss later on if you like. And you recognize these pictures as well. These 2 cows need complete different approaches when it comes to fertility. This is all infertility on the screen. Before the lunch, I linked this to inflammation, but now I'm one step further. If this farmer calls me to say I have a subfertile herd and I see this, then I turn my car and run away, right? This is infertility. This is infertility, or call it subfertility. So very important. This is also a shame that people ask me, show it a little bit longer. I don't have the time to show all the pathways. It's a paper published by Stephen LeBlanc's group in Canada. He is one of the greatest scientists in dairy cow management that I do know. It's published in Journal of Dairy Science. Great, very nice. If you want to have this overview, send me an email. I'm happy to send you the PowerPoint. All right. So how to stimulate that ovarian activity? How can we make these ovaries working and having the follicles growing and having the cow ovulating in time? Like at least 2 cycles before we want to breed the cow. That's the ideal situation. Well, first of all, try to manage and convince the farmer to manage for appetite and dry matter intake. Avoid too fat cows at calving. Feed bunks should always be available for fresh cows. Always. Top feed, top disease management. We spoke about it this morning. Climate and housing conditions, super important. And these transition cows, they do deserve super focus. Focus on glucogenic diets. Glucogenic diets in the early weeks postpartum will generate glucose in the cow. And that's the only trick we can have to uncouple or recouple the growth hormone IGF-1. You still remember? More starch, as long as it's not too much, of course, will lead to more glucose. More glucose will just push a little bit the insulin. A little bit more insulin will just couple the growth hormone IGF-1 axis in the liver, and everything goes well. Think about antioxidants or postpone the first AI. That's also an idea. But take care. If you postpone the AI because you think it's AI in cows at 80 days postpartum, why bother? Just wait 150 or 200 days after calving before AI. In our European systems, that's suicide. That's a ticking time bomb because the cows have too long calving intervals and they will risk to become too fat at the last part of lactation, leading to too fat cows in the next lactation, more metabolic stress, more infertility, more trouble. So, I spoke about this. I spoke about the importance of ovarian activity, linked to metabolism, linked to inflammation. Let me speak about the long-term history of the oocyte. Well, I'm showing figures of a study from a Brazilian that did his master thesis in the US, and he did fantastic work. What did he do? Don't look at the figures. I'll try to explain. He compared cows based on how they performed the first 3 weeks after calving. And he grouped the cows, he blocked the cows based on condition score changes. So what you see here on the top, you see cows losing a lot of condition early weeks postpartum, compared to cows that didn't lose a lot of condition or even gained a little bit of condition the early weeks after calving. So these cows, the red cows, are those who are in metabolic stress. I hope you agree. compared to those who were like easygoing, well-managed cows that were able to cover up, etc., and go for sustained milk production these first early 3 weeks. And then he followed up these cows for 3 months. And after 3 months, he superovulated all these cows, he inseminated all these cows, and he flushed the embryos out. And he looked at the embryo quality. At the moments of superovulation and insemination and etc., cows were not different in health status, were not different in milk production. So the only difference was the history, how the cows were behaving the first 3 weeks after calving. That should be very clear. And then he looked at the quality of what he flushed out of the uteri. And so I'm just dragging your attention to 2 major findings. First of all, the proportion of degenerated embryos. He saw that 1 out of 3 embryos flushed from the cows that had a troublesome first 3 weeks after calving, 1 out of 3 of these embryos were degenerated. That was absolutely not the case in the group of cows that had a very easygoing early postpartum period. They had good embryos. They had only very low proportions of degenerated embryos. Compared to the quality 1 embryos, look, only 50% in that treble cow early postpartum. 3 months later, only 50% of these embryos seem to be okay. That percentage is significantly higher in the other group. So the only difference that he could use to explain the differences in these embryo observations was the history of the cow, what happened 2.5 months, 3 months before. So yes, we have good reasons to assume that what happens early postpartum has a carryover effect, a kind of a memory lack over effect on how the cow performs at the moment of breeding. That's very important. However, these are associations. There are no causations. More data in that direction. If you look at large epidemiological data sets, you see here the pregnancy per AI. So chances to stay pregnant after AI, and you see these chances are significantly lower when 1 or even more than 1 clinical disease was described in the early postpartum period. So, okay, we have, based on figures, reasons to assume that there is a kind of a memory thing we have to take into account in our modern dairy cows. And there's good reasons to think in that direction, because as I said before, an oocyte takes about 3 to 4 months before it reaches ovulation. By the way, it's the same in human. A human follicle, it's about 110 days before activation, from activation, to ovulation. We are studying in the laboratory in a very intense way whether these dormant follicles or these early activated follicles, do they feel something? Or do they bother, or do they don't bother? I mean, we don't know. Nobody knows. That's what we're trying to investigate. We don't use human samples there. We don't use bovine samples. We use mouse samples. So anyway, but I'll stick to bovine. It was Jack Britt, he was a dairyman and an academic in the US, who said in the late 80s, I think that a follicle that is going through a troublesome cow will carry the consequences up to 3 months later. He hypothesized that. We call it the Britt's hypothesis. He visited my lab. We discussed about all kinds of protocols, how to prove it. So that's what we tried to do. And we designed a kind of an experiment trying to explore more about sensitivity of that little chapped follicle there waiting to be activated and eventually be stimulated to grow to pre-ovulatory status. So the question is, does negative energy balance, inflammation, oxidative stress, etc., affects the dormant follicle? So that's what we tried to do. What did we do? In a quite straightforward experimental design, we followed up cows on only multiparous cows because they are completely different from primiparous cows when it comes to metabolism. At week 8, these cows were all synchronized. And at week 8, we took follicular fluid samples. So that I will call it more correctly. So, or tell it more correctly. At week 2, so during transition, during a negative energy balance, we took a serum sample. We did the same at the moment of follicular fluid aspiration. So at week 8, we synchronized the cows and we collected follicular fluid and we isolated the granulosa cells, the cumulus cells. I started my talk with that juicy follicle, with the juicy oocyte and the supporting cells, you still remember? These cumulus cells are somatic cells in connection with the oocyte, and they support oocyte quality for 3 months long. So instead of looking at one oocyte, we decided to look at these million cumulus cells. They give us much more information, much more DNA, much more genes to evaluate, while if we miss to collect one oocyte, the whole sample is lost. So that's what we did. So at week 8, we screened the granulosa cell gene expression pattern with an RNA sequencing approach. Sorry. It's just a whole genome gene expression analysis. That's it. And we try to interrelate the gene expression analysis with the metabolic background of the cow many weeks before. And so we hypothesized that what happens here during transition can be read out here in the gene expression patterns of the cumulus oocyte complex at the moment of breeding. I was a non-believer because in this type of research, there is a lot of fog, factors that you don't control for. Anyway, I got a very, and he's still in the lab, a very stubborn postdoc. And he said, screw you, I will investigate it. So we went on with the study, luckily. So these are the cumulus cells and the granulosa cells that we focused on. But then, of course, I have to tell you a little bit of difficult stuff in that way. So at week 2 and week 8, we compared blood composition based on beta-carotene, vitamin E, antioxidants, NEFA concentrations, but many more comparisons. And we contrasted the cows that were high in these factors versus low in these factors at 2 weeks after calving. And then we took these cows 8 weeks later and checked their granulosa cell expression patterns. Are you still with me? Okay. So that's what we did. So for example, this is beta-carotene concentration at 2 weeks after calving. You have the lowest quartile 1 cows and you have the highest quartile 4 cows. We followed these cows and we checked gene expression differences in the oocyte supporting cells 8 weeks later. Okay, and then we need bioinformaticians to analyze all the data, and I'll try to summarize it. Look, these are the concentrations, or at least the factors that we analyzed in the blood at 2 weeks after calving. And you see these are the numbers of genes in the granulosa cells at week 8. So cows high or low in beta-carotene have follicles 8 weeks later with 190 differentially expressed genes. Wow, is that, yeah. If we check the gene identity, all these genes are linked to antioxidative processes, insulin sensitivity, gonadotropin receptivity. And then I think, whoa, we are in the middle of the war. It's very relevant what we see. Look at this. If we contrast cows that were high in beta-carotene and vitamin E versus low in beta-carotene and low in vitamin E at week 2, we see 340 genes differentially expressed 8 weeks later. So there we can say, well, these follicles, they have a kind of a memory. They act differently at week 8 when they went through another metabolic and inflammatory status just after calving. But then I see you thinking, what about their condition at week 8? Imagine that cows are high or low in beta-carotene at week 8. Probably that explains their differential follicular activity, right? Well, that was not the case. Look, only beta-carotene plays a role, but the genes were completely different in these two groups. So blood composition that was different in cows did not play a role in gene expression level at week 8. So based on these results, these are the genes we explored, and these are more genes that we explored. Based on these results, we can say that the behavior of this follicle, the follicle you and I are going to ovulate with our offspring protocols, the follicles you and I are going to use to inseminate the oocyte of, well, the behavior of these supporting cells are much more dependent on what happened early postpartum compared to what happened with these cows at week 8. So Jack Britt, he was right from the beginning. We showed it at the molecular level. There is good reason to assume there is a carryover effect of early postpartum health to the follicle health and oocyte quality much later after calving. So to conclude, These results provide evidence that low blood antioxidant and high NEVA concentration in dairy cows early postpartum have a long-lasting impact on the transcriptome profile of the granulosa cells, which is detectable in the dominant follicles. We could not attribute these changes to factors at week 8. Such long-term impact on dominant follicle physiology at the moment of breeding appears to be initiated during the early stages of follicular development, so during early lactation. So if you want to think about breeding, if we want to think about fertility, we have to take care of that cow much earlier. The effect of negative energy bands may be mitigated by optimized antioxidant concentrations before breeding. That's what we did. We normalized beta-carotene, and then we saw that all the differentially expressed genes disappeared. So we can play with that. Okay, that was published for those who want to revisit that paper. So Walid is my golden postdoc project coordinator person in the laboratory, and you see Jack was a co-author of this publication. So at the moment of ovulation, the oocyte will remember this and this, right? So please think about it when you speak about fertility management on dairy farm. Fertility managing means that you have to start early enough in lactation. And then to end up, I would like to highlight a little bit the high fertility cycle, which is especially in European conditions extremely important. It's a concept that also comes from the United States. It's like 5 years old, something like that. It's a very nice concept. It highly applies. It's nothing new. You see that cows that have a high condition at calving will have higher calving intervals. Is this new information? No. Cows with longer calving intervals have a higher risk to accumulate too much fat. And you know what it means now to be too fat at calving. So we can look differently at this. If we see cows with shorter calving intervals, they tend to mobilize much lower amount of condition in transition, while cows with longer calving intervals have higher condition score changes. And we all know that this change is for us so important. So if we want to keep the cows in our what we call an high fertility cycles from one lactation to another, to keep the cow in the best possible condition, we need to keep the calving interval short enough. Otherwise, we end up in disaster. So the high fertility cycle means that you should have cows having a calf again within 130 days after calving. So to summarize, low fertility leads to too long calving intervals. I hope you agree. Too long calving intervals leads to too high condition in the late lactation. At least in our Western European conditions, farms are too small to work in production groups, so they're all fat. The same TMR, early postpartum to late. So the last trimester cows, they just accumulate lots of fats. So too long calving intervals means too high condition score in late lactation. Don't forget feed bank management. If you have overstocking problems, you have feed bank competition. That's a disaster for transition cows. That's a disaster. But that can also lead to too high condition in late lactation for the dominant cows. Too high condition leads to too high condition at calving, more metabolic stress and negative energy balance, and more infertility. Don't forget, low fertility will keep the low fertility on the farm. I hope you agree. So just shooting with hormones to deal with low fertility, that's a symptom you're treating, which is important. Take away the symptom to make the problem manageable. But then you need to manage. Okay? Very important. And just lastly, if you say, well, we don't care about early first inseminations, we wait up to 200 days. In our settings, that tend to be quite dangerous because you end up with overconditioned cows and fertility problems just in the next lactation. So to summarize, we spoke about the sensitive oocyte at the moment of breeding. Be sure you breed healthy cows, or be sure you have healthy cows before you breed them. Second, how can we make that ovary resuming ovarian activity as soon as possible? Well, Think about insulin and IGF-1. Please don't forget it. And thirdly, what about the history of that oocyte? I hope I show you that the oocyte carry a backpack of history on her back. And so if you have a bad history, you can use off-sync protocols to ovulate this oocyte, but there will be no cough. I hope you agree. Good. So what is then the impact on your repro toolbox? If you really want to go on a farm, use this scheme, print it in the color diagrams and show it to the farmer. If you really want to sustainably tackle a fertility problem on a farm, think about genetics, heifer rearing. Look, it's there. We heard wonderful things about it today. We will hear more about it just in a minute. Think about the high fertility cycle. Think about the importance of the dry period. And think about early lactation. But you see, if you only start thinking about fertility and early lactation, you're too late. You're only firefighting. However, off-sync protocols are great tools to push a cow into pregnancy if there's no other option. So please use them to keep the calving interval as short as possible. With this, I still have 5 minutes, so I will read the conclusions very slowly. Healthy cows, they produce a lot of milk and can be perfectly fertile. But choose the best possible starting material. And I'm just referring to the heifer information we received earlier today from Given. Strong and high genetic merit heifers that are following a good growth path. Fertility management starts in the previous lactation. Transition success is programmed in the previous lactation. Don't forget it. At transition, you're too late to manage. Then you can only cure. Transition success is the key for optimal fertility. We think about 2 hormones. I still have 4 minutes and 10 seconds. IGF-1 and insulin. Great, I'm a very happy man. Care for the uterus and care for its optimal recovery. If you have a voluntary waiting period that ends at 65 days and you ask the farmer, is her uterus healthy? And when the farmer says, I don't know, then we have a problem. Off-sync protocols do not cover up for bad oocyte history, but they still are fantastic tools and they work perfectly to keep the calving interval as short as possible and to stay into the high fertility cycle. What's the biggest reason why an off-sync protocol is not yielding good results? It starts with a C. Compliance. That's a man-made problem, not a cow issue. Okay, so with this, I used this graph before, but now I say first beauty and health and then reproduce. Thank you very much.
Hi, do we have any questions for Prof? Any questions? Prof Holm.
Joe, those cows that have the biggest negative change in body condition score after calving compared to the ones with no change, are they not also the ones that produce the most?
Yeah, sometimes, but not always. That's absolutely true. And again, body condition score to a certain extent is a normal event. It happens in humans. It happens in a lot of mammals. Sea lions are our most clear example in terms of fat mobilization after parturition. But yeah, the higher producers tend to mobilize more. And if you manage them perfectly, they can support it. Don't forget that every molecule of fat that you mobilize needs to pass one most important organ, that's the liver. And so research nicely shows, and that's research that has been done 20 years ago, that the more fatty acids that are introduced in the liver, the more the nervus vagus is stimulated, goes up with information to the brain and just shouts out to the brain, stop eating. We call it the HOT theory. the hepatic oxidation theory. The more oxidation takes place in the liver, the more the switch off of appetite is threatened. So if you see, what I try to implement on farmers, and more it's happening with cameras and automated systems, is to follow condition from the moment of drying off, calving, which is not so easy to assess automatically the condition at calving due to the fact that, especially in the first lactation cows, as we heard this morning. And then 2 weeks after, 5 weeks after, etc. And then it's always very clear, those cows that follow the tree are those that having the highest condition score changes. So using milk production as an excuse is not right, because milk production is the result, is an end result. It's a symptom. Condition score loss is a symptom, right? Don't call it a cause. It's a symptom, it's a consequence. Do you get the shift? Yes.
Hi. Any more questions? Nothing. Okay. Thank you. Thank you so much, Prof.
Welcome.
Okay, then we will have a quick tea break, and I just want to ask you all to please be seated again by 3:30 for our final session. Thank you.